the guest speaker for today's session. I welcome Dr. Jeevan Duntulwar, Principal of our college, Dr. Reena Saha, Vice Principal and IQAC Coordinator, Dr. Sonparote, Head of the Department, Dr. Gazbe, Coordinator of Zoological Society, and all the staff members of the college and students. We have witnessed a wonderful lecture a day before, that is on 28th of February. And in continuation with the lecture series, we have our next speaker for today, who is Dr. Vinod Scaria. I welcome you, sir, once again on behalf of all the staff of the college. Then we know that we are celebrating this Science Day, but we could not organize two lectures on 28th, so we are meeting again today. So I would like to request Dr. Reena Saha to kindly brief us about the activities of the IQAC, and then we can proceed. Over to you, madam.
Very good morning, Dr. Scaria. This is Reena Saha from Mohta Science College. Audible? Am I audible, sir? Am I audible? So Nikhil has asked me to give the details of role of IQAC in quality enhancement. So my goal as a IQAC coordinator is to develop an action plan for the academic year to interact with the students, management, management, college and society to access the elicit suggestions, recommendations in important areas like introducing need-based academic program and improving infrastructural facilities and share the best practices with others to achieve objectives of IQAC. We have done MOUs with different NGOs. Their participation will be very much helpful for our students to be an entrepreneur, to be a good service provider, or to be a good farmer for sustainable farming. Another is to develop a campus ecosystem that will ensure the best of teaching and learning experience for the students for their all-round excellence. The IQAC organized a number of talks and workshops on quality-related themes for the teaching, learning, evaluation, and best practices for quality sustenance in higher education. Like we had arranged some program like attributes of research for PG students, right to information for non-teaching staff, then capacity building of teaching and administrative staff also. We took training workshop on self-defense and empowerment of international expert was organized to promote awareness about self-protection in girls. Most important function of IQAC is to develop realistic and attainable benchmark for each of the academic and administrative activities by fixing standards for all the activities. Our team is working diligently and good results are reflected. For improvement on students' performance in academic and placement activities, CRT programs are initiated for PG students and CHB teachers of the college by inviting very dynamic resource persons. And we are observing very good result and good performance of our students. Our website is also very much active and updated with institutional information and also for communication connectivity. So I have a satisfaction of having accomplished the academic and administrative target we had set for ourselves and spur to do more and more for the next season. So I conclude here as an IQAC coordinator. My team of PG faculty, my team of all the teachers of zoology department are working very hard to get the students updated, more and more information, and this endeavor of getting MOU with this reputed institution of CSIR was done by Dr. Nikhil Pandey. I appreciate his efforts, and now I stop here and I ask Nikhil Pandey again to take that.
Thank you very much, madam. Now I would like to take this opportunity to introduce today's speaker. He has a very impressive biodata, as it is evident from the list of publications that he has provided. Before going to his publications, I would like to introduce him as the principal scientist who is working at CSIR Institute of Genomics and Integrative Biology, located at Delhi. He is also the adjunct professor at Indraprastha Institute of Information Technology, Delhi. When you look at his bio sketch, he is basically a clinician turned computational biologist. His research interests basically lie in function, organization, and regulation of vertebrate genome and how genome variations could potentially impact them. Very importantly, he has been involved actively and he is the co-founder of the Genomics for Understanding Rare Disease India Alliance Network, that is abbreviated as Guardian. And I must say that we could not invite him on 28th because that is also the rare disease day. So he was busy in some other activity. But fortunately, we could invite him today and we will be able to hear his talk on the latest hot topic of today, that is the SARS-CoV-2 genome and what is the situation as far as India is concerned. Basically, Dr. Vinod Scaria is from Kerala and he has basically done his initial education as MBBS and then he switched on to the research field. That is, once he graduated, he then pursued research in genomics and information technology. He has also the member of various editorial boards, including very reputed journals like PLOS One, then Journal of Orthopedics, then Frontiers in System Biology, Journal of Translational Medicine, and so on. He has memberships to various international bodies as well as national bodies. He has received various awards and fellowships, including very prestigious Fellow of the Royal Society of Biology, then Cowley Frontiers of Science Fellow, US National Academy of Sciences, then CSIR Young Scientist, BioClues Innovation Research Development Award, and so on. I can just go on reading his biodata for a few more minutes, but I would like to highlight some more points of his biosketch. He has been actively involved in technology commercialization. As we have seen the other day, he was also involved in development of a commercial product, that is MitoSure, in 2016. Then in 2017, he was involved in developing tests which are now commercially utilized by Dr. Lal Pathology Lab. And in 2018, he has been involved in the know-how of interpretation of nutrigenetics data for consumer testing, and which has been now licensed to Adams Genetics Private Limited. Further, he also has a patent to his credit named as human microRNA targets in HIV genome and a method of identification thereof. When you look at his publications, he has 170 publications to his credit, and I am also fortunate to have been given the opportunity to become one of the authors of the recent papers, which was published very recently. With this elaborate introduction, I would like to conclude myself, and I don't want to waste much of the time. I would straight away request Dr. Vinod Scaria to kindly begin his presentation. Over to you, sir.
Thank you, Dr. Nikhil. Am I audible and can you see my screen?
Yeah. So your presentation is also visible and your video is also visible.
Right. Thank you very much, Dr. Nikhil, for organizing this talk and inviting me for this session. Over the next 30 minutes or so, I will briefly describe on the COVID-19 genomics in India and what we have actually learned from doing the COVID genomes. Now, before I start my talk, let me first introduce what we work on. We call ourselves genome informaticians, which necessarily means that we work on genomes using computers and computational tools. And we try to address a variety of problems in biology using computational tools. Briefly, my large proportion of my lab works in the area of non-coding RNAs and trying to understand how non-coding RNAs could regulate different genomic components. We also work extensively with clinicians trying to understand genetic diseases across the country as part of a network which we call as Guardian. We also work on population scale genomes. Now, there are a large number of countries who have population scale genome programs, including India, where we try to understand the genetic epidemiology of rare genetic conditions. And we also work on a bit on the pathogen genomes. We used to work on tuberculosis. And of course, with the SARS-CoV-2 pandemic, we have initiated work on the SARS-CoV-2 virus. Of course, you could find more information about the lab and the lab research at the lab website here. Now, for students, what you need to keep in mind is that traditional microbiology has underwent a quantum jump or a quantum leap with the advent of next generation sequencing. And this has been more evident in the current pandemic because compared to the traditional aspects of microbiology, where in a pandemic you would investigate the organism by culturing the virus or culturing the organism, isolating them in a pure form, and then in many ways confirming by molecular approaches or by microscopy. This pandemic has been very different in the sense that none of these approaches used to be or are the mainstay for investigating the pandemic. And this is very evident from the first genome that has been sequenced and made available by Chinese researchers early in the pandemic. This paper is available, published in February 2020, but the genome sequencing was available much early on. And everything that we do in terms of the genomics has been based on this initial genome sequences which have been made available by researchers in China. Now, if you go to the materials and methods section of this paper, what we find is quite remarkable that there was never a step of culture or identification or purification of the virus. What really happened was that the sample was processed, RNA was extracted and was sequenced and analyzed to understand the causative organism, not just the causative organism, the genome of the causative organism in the context of other genomes that have been sequenced and made available before. So in other words, traditional microbiology has now been replaced by a better microscope, and that better microscope is what we call as genome sequencing. Now, as we do genomes, one thing that you need to keep in mind is that the virus mutates, and the virus mutates at a pretty constant rate across time period and across geographies as it's spread across the world. And this mutation rate is approximately 1 in around 7 to 10 days. I will come to this number again in a few slides from now. And that is essentially the basis of all the genomics that we do in understanding viral evolution and viral genetic epidemiology. And this is sort of exemplified in this particular slide in here. These are 3 genomes from early in the pandemic in 2020. The first genome, as you see here, was isolated in December 26th in 2019. And then, of course, there are 2 other genomes which are for the purpose of comparison from January 8th and February 27, all of them from China. And what you see here is that over time, the virus has accumulated genetic mutations, and these genetic mutations are accumulated on a genetic background. So it keeps on accumulating mutations in a particular genetic background as it spreads across the country or across the region, or for that matter, across the world over the time. And this is really, really important to keep in mind, because practically everything that we do in viral genomics is based on this concept that the virus mutates and the mutation happens at a constant rate, and mutations accumulate over a background of genetic mutations which it already has. And now there have been a large push into making these sequences available, and that has been a conscious push and a quite significant paradigm shift, in the sense that there have been a large number of genomes today, amounting close to around 600,000 genomes. It is not one or two or even 1,000, 600,000 genomes that have been made available in public domain, the sources like the SAID or the GenBank databases. Now, that provides us an enormous amount of information which can be used to understand how did the virus evolve and how did the virus become or accumulate functional variants through the process of evolution. The other thing that we need to keep in mind is that as the virus mutates and as it transmits across the world or a region, as I said before, that the virus mutates on a genetic background. So assume that this is a chain of events where the virus mutated, accumulated mutation number 1, then accumulated mutation number 2, and at that point of time, it could accumulate mutation number 3 or mutation number 4, and then therefore there's a divergence in the chain. So at any point in time, if the virus is sequenced, then what you would realize is that the proportions of 1 to 3 and 1 to 4 are supposed to be the same, because the mutation rate and the spread of the virus is approximately the same across the world. But at some points in time, what you realize is that the virus, the proportion of a particular kind of virus, in this particular case 1 to 4, could be much larger than, say, for example, 1 to 3 or 1 to 3, 5. Now, what does this mean? It just means that there has been an emergence event, and this emergence event could be because of host factors. That means, for example, an individual who could have transmitted the infection to a large number of people, for example, in a congregation, or a virus which has accumulated a mutation or multiple genetic mutations, which allows it to spread faster and wider. So just having a large proportion of virus of a particular genetic lineage in a point in time doesn't necessarily mean that that is just because of viral mutations. It could also be because of human factors which spread the infection far and wide. So this is something that we need to keep in mind. The other thing that you need to keep in mind is that what we talk about are genetic lineages of the virus and not really strains of the virus. So when we talk about SARS-CoV-2, that is a distinct strain of the virus compared to SARS-CoV or the Middle Eastern Respiratory Syndrome or the MERS-CoV virus. And everything else within SARS-CoV-2 are essentially genetic lineages, or what we call as clades of the virus, each of them different by just a few genetic mutations and not significantly different from each other. So we should not really call them as strains of SARS-CoV-2, but rather genetic variants or genetic lineages of SARS-CoV-2. This is something that you need to keep in mind, because there's a lot of papers, newspapers, and public media which sort of suggest that a new strain of virus is emerging. They're essentially new genetic lineages or new variants in the genome of the virus which is emerging. Now, the global data can actually be put into a context, and this is available in a resource called NextStrain. You could browse this data freely. And briefly speaking, the virus lineages in the old classification of NextStrain has been divided into approximately around 10 lineages. And early in the pandemic, as early as Jan 2020, many of these lineages roughly corresponded to the geographical spread of the particular virus. Like what you see here is an A2A of this big, large group of viral lineages. essentially spread out of Europe, while the B lineages, which include B, B1, B2, and B4, are essentially viruses which spread out of China or into China. Of course, you would have these green dots in here, which is the A3, which essentially spread into Iran, and so on and so forth. So just by looking at the lineage of the virus or the genetic mutations which mark these genetic lineages, you would be able to say early in the pandemic from where did these infections come into a particular region. But of course, as it spread across the region in time and space, this information is lost. Now, if you ask the question about India, over the last 1 year, the first case was identified in January 2020, over now a year ago. And over the last 1 year, there have been close to around 9.7 million cases, and of course, 140,000 deaths in the country. And we have been collecting this genomic information and genetic variants in a very systematic format in this resource, what we call as INDICOVE. You can access this resource for free. And this data has been contributed by a large number of institutions across the country who have made available the genomes in public domain. And it's estimated that approximately around 10,000 odd genomes of SARS-CoV-2 have been sequenced in India, and approximately around 7,000 odd of these genomes are available in public domain, which forms the basis for this resource called INDICOVE. You need to also understand that to be doing genome sequencing at scale, you also need to have tools and methods to make genome sequencing faster, cheaper, and efficient. And one of these approaches is what we call as a COVID-seq. And the COVID-seq essentially is an assay which was developed by Illumina and trialed or clinically validated for the first time in the lab at IGIB. And this is spearheaded by Dr. Sridhar Shivasubbu, who would have heard his talk last weekend. He, with a fantastic set of young researchers, put together this assay in a clinical setting, and that's what we call as a mega lab. And the idea of the mega lab is to be able to create a platform which is flexible and modular in our ability to sequence genomes, and it can sequence anything starting from 96 samples to approximately 3,000 samples in one single run on a genome sequencer to be able to understand the genetic epidemiology of the virus and genetic variants in the virus as it spread across the country. And apart from the sequencing, this is also combined with an almost automated system to provide information about genetic lineages and genetic variations in the virus sequenced from the genomes. Sorry for the text in the slide, which is overlapping, but we have been fortunate to have been working with a large number of states, which includes Kerala, Tamil Nadu, Karnataka, Andhra Pradesh, Orissa, Rajasthan, and Western UP to understand the genetic epidemiology of the virus early in the pandemic. And what we found here was quite remarkable. And if you put the data that is available, sequenced by us, as well as sequenced by other laboratories in the context of a phylogenetic tree, what you see here is that compared to the 10 odd clades that were prevalent across the world, India has 6 out of those which maps perfectly in the genetic lineage and the genetic mutations which mark these lineages. And apart from that, as you see here in purple, there's a distinctly large cluster of the virus which doesn't have a particular large lineage mapped anywhere else in the world. So then, therefore, we map this lineage, and we call this lineage as IA3I. I to signify that it is an Indian lineage predominantly found in India, but not exclusively found in India. A3I to signify that it has been a lineage that has diverged out from the A3 lineage of the virus. And A3 lineages of the virus, as I said before, are the viruses which came out of Iran. Now, while we still do not have an epidemiological understanding about how it has emerged out of the A3 lineage, of course, I will come to that in a few slides from now. But what you see here is that each of these genetic lineages could be marked by very specific genetic variants. And the IA3I lineage was marked by 4 genetic mutations, which are significantly distinct from the other mutations or the other variants that other lineages have across the world. One of these genetic mutations, not absolutely, but in a significant large proportion of the virus, was shared with the A3 lineage, therefore sort of suggesting a name as A3I, not to suggest that it emerged out of the A3 lineage, but it is very similar to the A3 lineage by one genetic mutation. And of course, what you could do is to also look at very significant properties of the virus. And one of the properties of the virus is its ability to accumulate genetic mutations. And that's essentially how the virus evolves. And what we show here is that the A3I or the IA3I lineage of the virus has a much lower mutation rate compared to the other viruses or other genetic lineages in India. So it is sort of expected that this virus lineage would at some point of time die down because it would be overpowered by viruses which have much more efficient system of evolving by accumulating genetic mutations. And that's what we see here, the bottom right panel, the purple color has diminished over time, and it has been taken over by the now predominant lineage, which is called the A2A lineage. Now, of course, you could look at the distribution of these lineages across the different states in the country. And what you see here is a map from sometime in September. Different states of the country has different proportions of the IA3I and the A2A lineages of the virus. The IA3I is in purple and the A2A is in blue-green. Now, different states have different proportions. This proportion could be large in states like, for example, Delhi, in, for example, places like Karnataka or Telangana, but it could be probably absent in places like, for example, Kerala. And this all depends upon how did the virus and the lineage spread across different parts of the country. And the size of the bubble is representative of the number of genomes that has been sequenced from each of the states. And of course, that's been very disproportionate. There have been large number of genomes that have been sequenced from Delhi, for example, large number of genomes that have been sequenced from Gujarat, with the Gujarat State Biotechnology Institution, a large number of genomes sequenced from, for example, Kerala. That is because it has a surveillance program in place, so on and so forth. And looking at the divergence of the virus, there's a central panel out here. What you see here is that the virus seems to have diverged from a central point clustered in space and time. And after that, it has spread across different parts of the country and in some cases, different parts of the world from this central event. So such kind of a map suggests that such a spread is essentially part of a super spreader event. And so what you can safely assume is that the virus spread after a super spreader event into different states of the country and in some cases different parts of the world. And just looking at the mutational origins of the virus and the background of the mutational origins, you can actually also map how the virus has got transmitted from city to city or state to state by interstate travel before and after the lockdowns have been implemented. And the thickness of these lines essentially suggest how close or how constant has been the influx of individuals between states who have contributed to the spread of the virus. And like here, you would see how the infection could have been contributed from, for example, Delhi to Telangana, from Gujarat to Orissa, and so on and so forth. So that's one of the other applications that we can build maps of molecular contacts of the virus to understand how did the virus spread across different parts of the country over time. And this is very useful to understand in our ability to contain the virus by appropriate measures. You could also create spatial maps of clusters of the virus. This is what we call as haplotype maps. And these are essentially built upon combinations of genetic variations. And such combinations of genetic variations could enable us to understand clusters of the virus spread across different regions of the country. And each of these large clusters are essentially outbreak events or super spreader events, which has happened at different points in time and in different regions of the country. And the proportion of the size of the and the circle signifies the number of genomes which is part of the cluster. The color signifies the state which has contributed to the cluster. And as you see here, a significant number of genomes are part of one or another cluster, and each of these clusters are essentially large events which are limited to one state. For example, like what you see here in Andhra Pradesh, or could encompass large number of states, like for example, this one, which is being contributed by multiple states. So in other words, you could understand how early spread and transmission of a virus has occurred in clusters of super spreading events which were spaced across the country. And of course, by doing genomics, you could understand many other things, which includes how to develop better diagnostics, how to understand the immune epitopes, which is very useful to build better vaccines. And one of the applications, of course, is to build better diagnostic reagents. And I'm sure all of you would know that the gold standard for diagnosis today is real-time PCR. And real-time PCR is based on the principle of PCR. So you need primers, which are very specific and they bind very specific to a genomic region, amplify a small section of the genomic region, and of course, which is probed by a fluorescent labeled oligonucleotide. Now, the amplification as well as probing is based on the specificity of the interaction between the oligonucleotide and the genomic material. And therefore, genetic mutations in either the binding sites of the primers or the probes could affect the efficacy of these reagents. So what you could do is to collect all primers and probes from across literature, from across package inserts, from across websites, and put together this information in the context of genetic variations and ask a very simple question: can genetic variations, which are very frequent in a particular population, map to these primers and probes? So what you see here in the panel is essentially the genetic variations. The y-axis is the frequency of these variants in the population. And if it is red, that necessarily means that it is in a primer or a probe binding site, which could affect the stability of interactions. You could calculate them using standard thermodynamic equations. And this entire work of putting together the primers and probes from literature and many other sources were done by a bunch of college students out of Bangalore who ventured out by volunteering to collect this information during the lockdowns. So that is one example of how a large number of students who are very cognizant of the fact of molecular medicine could now contribute to quite meaningful research in trying to build better diagnostics. And of course, in the clinical situation, or when you talk about the immunology for SARS-CoV-2, what we need to keep in mind is that we have two arms of the immune system. One is the humoral immune response, which is modulated by B cells, which produce antibodies, and you have a T-cell mediated immune system, or the cell-mediated immune response, which are mediated by T cells, which causes cytotoxicity. Now, in clinical situations, there are also very interesting problems which people come across. There are people who have persistent infections. In other words, they turn out to be positive for a much longer period of time. There are people who get multiple infections, either with a new virus, which is called reinfection, or with the same virus persisting after a period of time, which is called reactivation of the virus. Now, the only way to prove such events in an individual is to sequence the virus from these two episodes and understand are they similar or are they different. And such events could come only if you have a constant testing protocol. And one of these testing protocols were implemented by a collaborating institution by Dr. Vivek Gupta, who's a close colleague and collaborator, where all the healthcare workers on COVID duty were put on a regular surveillance once in every 15 days. So this allows us to get an idea of how did the infection progress in a particular healthcare worker community, and also understand very pertinent things like reinfection or reactivation in a close cohort of individuals. And this is one such case where two healthcare workers turned out to be PCR positive on regular surveillance. They were not symptomatic. They turned out to be PCR negative at a later point in time after being hospitalized and followed up, or after being in quarantine. And again, to test positive after a significant period in time. And the basic question was, is this a case of reinfection or is it a case of reactivation? And the only way to prove that is to sequence the virus, and therefore we could sequence the virus between episode 1 and episode 2 for the two individuals, naming them as I1 and I2. And what you see here is essentially the genetic mutations, which are significantly different between E1 and E2, which now suggests that E1 and E2 were caused by two distinctly different viruses of SARS-CoV-2, different by just a few genetic mutations. So this therefore becomes one of the first reports of asymptomatic reinfection. And mind you, such cases of asymptomatic reinfection can only be identified by closely putting individuals on surveillance. Because if you're asymptomatic, the individuals would never realize that they have SARS-CoV-2 infection, and that is true for 85% of the individuals out there who would never be clinically positive. But if you do an Rpa Pcr, you might find the virus infecting them. And of course, the other close mimic for such a situation is what we call as reactivation. And in reactivation, you expect the virus to be exactly the same between the two episodes of infection. And this is one such case, a child with stage 4 neuroblastoma who was on chemotherapy. Testing was done as part of routine surveillance before the dosage of chemotherapy, and the individual tested positive to test negative after some period of time, again to test positive after around 3 weeks, after around 42 days. Sorry. The virus from both these episodes were approximately the same, with no distinct variations which were different between them, and this sort of suggests that this is a case of viral reactivation rather than reinfection. And this therefore becomes the first case of a genomically proven case of viral reactivation in a patient with chemotherapy. And as I said, virus just evolve. One of the things that we need to keep in mind is that the virus elicits an immune response, and that's also what the vaccines elicit in a much controlled way. There are mutations now reported, and there's a growing literature, a corpus of literature on genetic mutations in the virus, which could escape one or more of monoclonal antibodies. There are 146 odd genetic mutations now reported, and individually these mutations are not of concern, but what is really of concern are multiple mutations of the same kind occurring on a virus. Now, except for a few genetic mutations like E484K or E484Q, which can escape multiple genetic multiple antibodies, individually, this is really not of much concern. And of course, for students out there who want to learn more about this, there is a very systematic resource of immune escape mutations, which you can actually browse through and understand the evidence on immune escape for each of them. What you need to also understand is that immune escape is essentially one of the Achilles heels for the SARS-CoV-2 virus. That is because the immune escape mutations largely cluster on the spike protein, because there is the exposed protein against which the immune response acts upon. And this also overlaps with what we call as the receptor binding domain of the virus. The blue colored region of the spike protein is the receptor binding domain of the virus. So the virus necessarily cannot mutate at all these positions without significantly losing out on its ability to latch to the human receptors. In other words, there is a balance that needs to be struck between its ability to identify and latch onto the human receptors and its ability to evade the antibody-mediated response, or what we call as the immune escape phenomenon. And of course, depending upon the ability to escape antibodies, whether they have one antibodies or they have multiple antibodies, they could be clustered at different regions in the spike protein. And this is something of great relevance because this information is very useful to identify and also look at viruses which are emerging, and also our ability to develop new vaccines which can counter this phenomenon of immune escape. Now, many of these genetic variations are very frequent and present across global populations, and there are at least half a dozen of them which are quite significantly present, more than 1% in at least one of the countries out there. And coming to India, also we do have immune escape mutations of big significance as N440K, which was found very prevalent in the southern state of Andhra Pradesh and now found in almost 10% in many other states, including Kerala. And very recently, there is another immune escape mutation, which is E484K, which is not part of this analysis, but appeared much later, which has been found in at least 2 genomes from Maharashtra. And now we know that there are many, many more genomes with E484K, which has been sequenced from India. So these are genetic mutations which we need to watch out. We don't need to panic about it, but as they emerge, and at least E484 has a capacity to evade multiple monoclonal antibodies and also reduce the efficacy of vaccines. So what we need to keep in mind is to sequence and identify the emergence of such viruses as they spread across different parts of the state, different parts of the country. And as we give out vaccines to different high-risk individuals, including healthcare workers, including now people over 60, and now also people over 45 with comorbidities, it's very, very important to have a genomic surveillance program where individuals who have been vaccinated but nevertheless could have breakthrough infections or reinfections could be put in surveillance, because this is a useful approach to identify what are the genetic mutations in the virus which would now evade the immune response. And also collection of a systematic database of immune escape variants and how do they emerge across the global populations is something that is of great interest, not just in academic interest, but also in our ability to control the pandemic by controlling the spread of the virus by other means, which includes better masking, better healthcare communication, and of course, washing the hands. And before I stop, let me acknowledge a lot of people who made this possible, a lot of people who collaborated. Most of the sequencing, almost all of the sequencing was done by Sridhar Shivasubbu's lab with a fantastic set of young researchers, fantastic set of collaborators from across different states in the country, and of course, students in the lab who contributed, despite the lockdown, to a large analysis of these data sets together. And of course, there are now multiple publications which have come out of it. And not but least, thanks to all the researchers who put their data sets in public domain from India and also from across the world. And all of this is possible with funding, and we acknowledge funding from CSIR, which has been consistent throughout the pandemic. I think I'll stop there and take questions, but before that, let me also introduce a suite of resources which students can access and understand more about the SARS-CoV-2 genomes in India and across the world. I think I'll stop there and take questions. Thank you.
Thank you very much, Dr. Vinod. It was indeed a very nice, elaborate presentation. And we could get some insights into what is happening at the genome level of coronavirus. Because we keep on reading in newspapers, I mean, I'm talking about students, they know that new strain has come, but what is actually happening at the genome level is not clearly very much understood. So thank you for giving us deep insights into what is happening at the genome of this corona variant or the mutations that are taking place in the genome. Students have not asked any question yet, but I would like to ask one question.
Sure.
Like when you have seen that there are reinfections in some patients, you have looked into genomes of coronavirus variant, but since you are also part of human genome sequencing project, have you seen, is there any correlation between the genome of patients who are suffering from reinfection and those who are not? Have you tried to compare anything like that?
Yeah, so we aim to compare that. So we have collected this, the genome, the blood samples of the individuals who had reinfections, but we have not yet sequenced. We are still running in single-digit numbers. We are not still crossed like a dozen cases.
Sir, you are not audible. You are muted, I suppose.
Oh, thank you very much, and sorry I got muted. So you asked a very relevant question, And yes, we do have collected blood samples from all these cases of reinfection with the idea that we'll be able to sequence. But unfortunately, these numbers are still in single digits, but not yet even crossed like a dozen samples. To do such kind of analysis, we need quite some number of samples. We are still open because reinfections are still happening across the country. The real limitation is that the samples from the first and the second episodes of infection are not readily available. But having said that, that is a limitation across the world. We are still open to taking up more cases of reinfections as they occur, and more importantly, to also take up cases of infection after the second dose of vaccination. So this is what we call as breakthrough infections. And the idea is that these individuals would have some defect in their immune mechanisms, and that's essentially why a virus could infect them for the second time, or at least one case, which is not proven, but of course in press, which has been reported for the third time in case of infection.
Okay, and what could be the reason behind having different periods of immunity? Like some of the individuals have antibodies for some days, some of them have for months. I mean, is it also related with the genome, or is it related with the variant of the virus?
Yeah, we don't really know and understand that in great detail. At least the experience from South Africa suggests that at least one variant, which is E484K, can cause reinfections in a significant number of individuals who have previously been infected. Now, apart from that one single report, there have not been much of studies about reinfections. So globally, if you look at the numbers, we have just crossed 60 cases of reinfection reported globally. And that's a very small number to start looking at patterns emerging. But having said that, at least of the 3 cases of reinfections that we have sequenced and reported, there are at least 2 cases where there is one variant, which is N440K, which is present in the case of reinfection. So these are essentially suggestions, but this still doesn't prove that the variant causes.
Right. And I have one more question. Like when we see comparative data from different states, in Maharashtra, I could see that there are more than 1,000 samples that you have studied, and almost out of total 19 variants, many of them or maximum number of them have been found from Maharashtra. Is there any some relation with the population genome, or it is due to the presence of cities like Mumbai where we have maximum visitors from outside? What can be the reason? I mean,
so there could be multiple.
The proportion is very small.
Right. So there could be multiple reasons. Okay. So first, of course, early in the pandemic, the number of variants is almost directly proportional to the number of introductions, or how many number of travelers have come from different portions of the world or different parts of the world. But as the pandemic progresses, what if a variant emerges, that necessarily means 2 things. As I said before, one is a host factor, which are individuals who end up spreading to multiple large number of people, and then therefore it spreads. But nevertheless, that doesn't change the mutation pattern. What really changes the mutation pattern are 2 things. One, time, right? So if you prolong the pandemic for quite a significant long period of time, that is enough time for the virus to accumulate more and more and more mutation, because there's one mutation approximately every 7 to 10 days. The second thing is essentially the treatment protocols which were put in place early in the pandemic, which allows the virus to mutate and accumulate very specific set of mutations which could escape or evade such therapeutic responses. So this could include, for example, drugs which have been given in the treatment. This could be, for example, convalescent plasma which was given during the treatment. This could be, for example, monoclonal antibodies which were given for the treatment. And in the future, this could also mean vaccines which are given to prevent SARS-CoV-2. So as vaccines become commonplace, we should also expect at least some of the virus to start evading the vaccine-mediated immune response by injecting the patients.
Right. So still, I mean, there are many things to explore now, because we don't know where this is heading to and when will this end. Probably this will not end, but the corona will settle down in the population like we have seasonal flu. Will it be the case, or what can be the future of this?
So yes, there are a large number of pandemics that humankind has handled before, which includes the influenza pandemic. That doesn't mean that the human race has been eliminated from the face of the earth. Viruses are smart, but humans are much more smarter.
Thankfully, yes. Anyway, thank you very much. I would like to hand over this to Dr. Badiye if there are any questions from students. Madam, can you ask some questions?
First of all, very good afternoon, Dr. Scaria. It was very nice presentation. I must say the species involved you just told about, camel and human and bat. So, sir, I would like to ask, there is any other species which is under consideration of this coronavirus? Apart from this?
Yeah, so coronaviruses are in fact a very common set of viruses which causes very less symptoms like common cold. Coronaviruses are very prevalent across human populations, but that doesn't mean that they become a pandemic or they create a lot of mortality.
Yes.
Now, what has really happened are essentially zoonotic coronaviruses, which are otherwise very prevalent in the animal community, which has jumped into the humankind. So whether it is camels causing Middle Eastern coronavirus or bats causing the SARS-CoV-2. So these are essentially zoonotic viruses which has mutated so that they can jump the specificity. And these mutations typically occur in the spike protein because that is how they jump. And to jump, you need to realize that it now needs to recognize both the receptors of the host, where it is bat or pangolin for that matter, or the receptors of human, which is in this case ACE2. Now, what we need to keep in mind is that as humans interact closely with animals, right, and that has increasingly happened because of human factors, like we are destroying forests, we are destroying and engorging into the livelihood of other animals, and that necessarily means that these animals would now have a larger contact with humans than before, right? And of course, that now we travel quite extensively, that will take the infections far and wide. And this pandemic has essentially shown us that just a virus jumping from a host to another host doesn't really cause a pandemic, but it causes a pandemic once the individual who has been infected can carry it across a particular district, a particular state, a particular country, or across the world. Now, what you need to keep in mind is two things, right? To prevent future pandemics, we need to keep a close watch about zoonotic diseases or animal diseases, which are likely to jump into humans. This is not just coronaviruses. This also includes the Zika virus. This also includes Nipah virus, which all are zoonotic viruses which can jump into humans. And the second is we need to have a surveillance mechanism and a testing mechanism where we could identify the early cases and control the pandemic from spreading. Right? Like you have seen before that we had 2 rounds of Nipah outbreaks, at least in southern states of Kerala. We have multiple zoonotic virus outbreaks almost occurring annually in the country. Any one of them, if they are left unchecked, could become the next pandemic.
Thank you so much, sir. And we must have to watch on zoonotic animals for the coronavirus and other pandemics. Sir, there is a question from our student named Anuj Admane. The question is, how does climate change affect COVID-19 genetic makeup?
No, the climate change doesn't change the genetic makeup of the virus. What changes is essentially that humans and animals come into close contact. Either we eat those animals or the animals try to bite us. And that is because of close proximity, which was not there before. And climate change essentially creates an environment where this proximity is, or this contact is much more frequent, right? Like we cause afforestation, we cause climate change. That means that some regions of the forest is lost. The animals have to move out, and when they move out, they come into close contact with the human kind. And these animals are very simple animals that we come across. This could be bats, this could be monkeys, this could be anything. This could even be mosquitoes.
Students, any more questions? Thank you, sir. Over to Nikhil, sir.
Thank you, Dr. Badiye. I would like to personally thank Dr. Scaria for such a wonderful lecture and an interaction which was very thoughtful, and you gave us many insights into what is actually happening on the field. Because what we know is just from the reports that come to us, but you are actually working. You and your team is actually involved in this, working on this during this Corona pandemic. So you are no less than a soldier for us, and also I would like to thank for guiding us through this the scenario that is actually there on the field. So with this, I would like to hand over the mic to Dr. Sonparote for formal vote of thanks. Madam, over to you.
Okay. So good afternoon to all. It gives me immense pleasure to deliver the vote of thanks. On behalf of Zoology Department, I, Dr. Uma Sonparote, first of all like to thank our today's guest speaker, Dr. Vinod Scaria, who honored this event by sharing his valuable knowledge. And also thankful to CSIR IGIB New Delhi for making this event possible. We are grateful to all the members of Nagpur Shikshan Mandal to provide all the facilities. I thank our principal, Dr. Jeevan Tontulwar, for encouragement and all the necessary assistance. I am extremely thankful to our vice principal and IQAC coordinator, Dr. Reena Saha, for guiding us. I am very much thankful to Rahul Boyer for his gracious presence. I extend my thanks to all the faculties and support staff of our department for their support. Dr. Nikhil Pandey has played an appreciable role in the organization of this event and making it successful. I thank him for his great efforts. Lastly, I acknowledge the presence of all the teachers, BSc and MSc students for attending this talk. I hope your support in future events too. Thank you. Over to Nikhil, sir.
Thank you very much, madam. Dr. Vinod, we are hoping to hear you once again once you are comparatively free from the current work, and we hope to continue this association with IGIB also. So thank you very much once again.
Thank you, Dr. Nikhil, for this fantastic session, and thank you very much.
Thank you very much. Now I declare that the event is over. I thank everyone for joining. Have a nice day.